Prevent SARS3 video series continues. This is the 5th video, and I continue to explain the simplified model of coronavirus adaptation to human. So previously I had talked about the virus. This is the first detection or adaptation process of a natural bat coronavirus to lung or the human or pangolin intermediate host. So the virus was it was detected by the receptors prior to reaching its receptors, and it will be dragged into the cells and processed. So after this detection, the receptors pull the coronavirus within the cell, and inside the cells, a pocket process the virus with different enzymes, with different mutagenic agents, and the virus is really treated to be destroyed by the immune system. So this thing did not happen to SARS coronavirus, to this exact situation. Therefore, the coronaviruses, let's say, remember the first model. Initially, I had talked about 1 million virus inhaled by pangolin or other animal, and then they reach to the surface of the cells, and then from this situation, some of the RNA fragments, or some of the RNA with damage managed to escape this digestion process. So this is how coronaviruses naturally adapt to human cells or the intermediate host, and it doesn't happen without an attack, without the damage. And after this digestion process, the escaped viral RNA, coronavirus RNA of the bat, with heavy damage, finding its way on the ribosome, and the coronavirus, they have some enzymes, proteins, fixing their DNA. It's fixing its DNA. It's forming its new original adapted virus with genomic damage. That's the signature event in bat coronaviruses during their adaptation to human, except SARS coronavirus 2. So this is the model. This is the simple model. And the next round of infection, we will see the coronaviruses infecting the human cells with, in some cases, no sialic acid activity, so receptors cannot detect them, or with sunken, or the sialic acids are kind of buried below the surface of the virus, except SARS coronavirus 2. This is, I have to stress the situation. And actually, there is a 30-year-old hypothesis by Rossman. Its name is canyon hypothesis or cavities hypothesis. In this, and this situation is canonical in almost most of the human viruses. But the other indication why SARS2 is not a normally adapted virus, because it's homology with its parent bat coronavirus, is 97%. So this indicates that SARS2 did not pass through this digestion, detection, this natural process, harsh treatment in pangolin, camel, alpaca, doesn't matter. It doesn't pass through this process. So I finalized the model. In the next videos, I'm going to talk about, based on the genomes of 6 coronaviruses prior to SARS2, and I will show you over the genomes the intensity of this damage, genomic damage. Thanks for watching.